Jiodong Gao from Rice University, and he is going to talk about marine particulate matter. And his title is Using Solid-State C13 NMR to Understand the Source and Transport Mechanisms of Marine Particulate Organic Matter, or EOC. So, go ahead.
Good morning, everyone. This is Xiaodong Gao, and I'm speaking from Houston, Texas. And thanks for organizing this great workshop and for the invitation. Only wish I could be there in person. I just got back from a field sampling trip in Arkansas. I'd rather be at the beach instead of in the woods. First, I would like to thank my co-authors, Megan Wright, an outstanding undergraduate working with us for this project, and my boss, Carrie Marcelo, who is also online with us today. And okay, Azmarat just gave us a great overview of the history and application of NMR in soil science for soil organic matter. And Adriana further discussed using NMR to tracking roots decomposition in soils. And now I'm going to talk something different, using NMR to study marine carbon, more specifically, marine sinking particulate organic matter. And I have to admit, I'm also trained as a soil scientist, not an oceanographer. I also love soils, but this has been a fun learning experience for me. Okay, the outline of my today's talk. I assume not everyone in the audience is an environmental scientist, so I will give a brief introduction of the global carbon cycle and the role of ocean, followed by a marine biological pump and global research effort to deploy time series sediment traps to collect the sinking particulate organic matter to study marine biological pump. Then I will let you know, explain the NMR instrument we're using and the pulse sequence we're using, and a brief introduction of the POC sample locations where we received those samples, followed by objectives and the key research questions we would like to answer, and finally results and conclusions. Next slide, okay. Okay, here we go. And this figure shows the most up-to-date global carbon cycle, the major reservoirs and the annual flux. You already probably saw some of this in Azmarat's talk and Adriana's talk. I just want to focus on the role of ocean. Today's atmosphere contains around 890 gigaton carbon. And the soils combined with the vegetations contains more than double of the total carbon in the atmosphere and holds great potential for carbon sequestration as mentioned by Azmarat. But they are small compared to the size of ocean. If you look at the area of which indicates the carbon reservoir, ocean is actually the largest active carbon reservoir, which contains more than 38,000 gigaton carbon. That is more than 40 times more than the atmosphere. Most of the carbon in ocean is in the dissolved inorganic form, with some portion in the dissolved organic and particulate organic form that are the most biological active. In addition to that, ocean is also the largest negative carbon sink. It takes around 1/3 of total carbon released by fossil fuel combustion each year. And a large portion of this carbon is absorbed by ocean, is transported to the deep ocean by the marine biological pump, which I will discuss in more details in next slides. Okay. Marine biological pump is a set of physical and biological process, which includes the gravitational setting pump and the vertical migration pump of zooplanktons and the physical mixing pump by ocean currency. The marine biological pump transfers the fast cycling carbon in the sunlit surface to the slow cycling carbon in the ocean's interior. And each year, biological carbon transfers around 11 gigaton carbon into the ocean's interior globally. And noted that this is more than the total carbon released by fossil fuel combustion each year. POM is the hotspot for microbial activities, which is rich in carbon resource and nutrients. And the dynamic of POM, which includes the source, chemical composition, degradation, and transport, will determine the transfer efficiency of biological carbon. With how much carbon is exported to the deep ocean for long-term storage versus how much carbon is remineralized back to CO2. Okay, global efforts such as the joint global oxygen flux study has deployed the long-term time series conical sediment traps all over the world at various steps to measure the rate, quantity, and the chemical property of sinking POCs to study the biological pump. And this decades-long studies has greatly improved our understanding of the transport mechanism of POC. This sediment samples from this POC samples from the sediment trap has been extensively studied, and the rich data set such as the flux, organic carbon, nitrogen, aluminum content, and delta carbon 13, delta carbon 14 exists for this POC samples. However, surprisingly limited NMR study has done for this valuable POC samples. NMR measurement can directly report the chemical composition of the POC, and we think NMR on this decades-long archived marine POC samples will improve our understanding on the source and the transport mechanism of POC in marine systems. Okay, we used an older Brook 200 MHz solid-state NMR for this study. Even though the older unit 200 unit has less sensitivity and low resolution compared to the more than high field NMRs such as 400 MHz or 600 Hz, it still works great for us, especially for the soils and sediment samples due to its high paramagnetic tolerance. Also, NMR is a non-destructive technique. No treatment is needed. This is especially valuable for the POC samples because we have so little subset samples materials available. Sometimes we do not have enough materials to fill the 4 mm rotor, and we have to use a spacer. For quantitative NMR analysis, the direct polarization or the DP is the golden standard. However, the quantitative DP analysis is almost feasible for large number low organic carbon POC samples, usually in the percent of 2 to 10% range. And it will take weeks to get a good, just one good DP spectra due to the extremely long recycle delay time. CP is faster, but less quantitative, the standard CP, because it's often underestimated certain functional groups due to their difference in proton proximity and polarization transfer efficiency. The newly developed multi-CP pulse sequence by Duan and Schmidt-Rohr used mainly short CP pulse CP blocks separated by recycle delay, and which allows signals to transfer more evenly across the different functional groups for better quantification. This development in the multi-CP made it possible to obtain large quantitative NMR data for the POC sample. Furthermore, as Marata mentioned before, we used an end-member molecular mixing modeling to get the more quantitative information of the chemical composition of POC. We used the carbon-nitrogen ratio of a sample and the NMR signal the signal distribution across the 7 predefined spectral regions, including LQ, NLQ, OLQ, di-OLQ, aromatic, phenolic, and amino carbonyl groups to calculate the relative abundance of each biochemical class, including carbohydrate, protein, lignin, lipid, char, and also a tuning parameter, carbonyl groups. Okay, we received time series thinking POC samples at various steps across the globe from our collaborators, including the Station W, which is located in the Northwest Atlantic. The Station W is one of the longest and oldest sediment trap studies led by Woods Hole scientists who are really the pioneers in the field. Also, we have the Station M, samples from Station M is led by the Montreal Bay Aquarium Research Institute. The Station M is located in the northeast Pacific, which is not far actually from the meeting site you guys are. Station M is also one of the longest sediment trap sites. And we have 3 additional Jacobs sites, including the Arabic Sea, the Arabic Sea here, and the Equator Pacific and the South Pacific, and as long as the East Japan Sea site. And our global NMR study on POC will report spatial and temporal changes in the bulk POC chemical composition. And to best of our knowledge, this is the first attempt to assess the global pattern of POC chemical composition using carbon-13 NMR. Okay, the objective of the study, we want to examine the temporal and the spatial changes in chemical composition of the POC samples during its transport from the ocean surface to depths, and to investigate the impact of geographic locations with different depositional environment on the mechanism of POC preservation and transport. During its transport from the surface where the POC is produced to the ocean floor, majority of the POC will be luminarized. Only about 1% of the POC will reach the sea floor and be buried for long-term storage. A key question we want to ask is, what happened to these POC samples during its sinking? Is the POC non-selectively preserved, which means chemical components of the POC is eaten by the microbes non-preferentially, or it's selectively preserved during its sinking to the deep ocean? For the only 3 NMR studies we can find in the literature for marine POC, unfortunately, they reported contradictory results. Hedge et al. Compared the NMR spectra of fresh phytoplankton and the POC samples at 2 different depths. They reported that the 3 spectras are very similar and concluded a non-selective preservation of POC during its sinking. On the other hand, Jimshik Huang reported that alkyl and aromatics were preferentially preserved during sinking, whereas Lu et al. Reported that the large heterogeneity in the surface POC chemical composition, which became more homogeneous at depths. I think all these inconsistent results can be explained by geographic variations, difference in depositional environment, and the supply of the organic matter. All these previous studies are one time or one single location measurement. I hope our global assessment will provide more insight for the POC preservation mechanisms and answer this key research question. Okay, another key question we want to ask is, can we use the LQ-OLQ ratio, which is a well-established index for soil organic matter decomposition, to assess marine POC decomposition? For soil organic matter, with the progress of decomposition, the OLQ, which is the major component of carbohydrate, were depleted first, with the enrichment of the LQ functional group, which is the main component of lipid. The ratio results in an increase in LQ-OLQ ratio with time. However, we know the marine carbon and the terrestrial carbon are very different. First, the organic carbon source materials are very, very different. For example, the left figure shows a representative spectrum of marine phytoplankton and terrestrial plant switchgrass roots. As we can see, they are very different. Switchgrass root is dominated by lignocellulose, where the lignin is unique to the terrestrial plants and the soil organic matter, whereas the phytoplankton is dominated by large lipid peak, the LQ peak, and the proteins. In addition, marine has a unique vertical migration of zooplankton, such as krill and copepods, which is rich in lipids in their biomass. It's reported that vertical migration of zooplankton can contribute as much as 10% to the total biological carbon pump. And third, soil organic matter and marine POC has different environmental conditions. For example, POC will lack the mineral protection and limit in oxygen exposure time. We are wondering if the LQ-OLQ ratio as the index of decomposition is still valid for marine POC, despite all these differences. Okay, results. For station W, we were able to obtain a whole year POC samples at all three depths. In this case, we can examine the seasonal change in POC and the change in chemical composition during its sinking from the surface to deep ocean. The previous research suggested that the station W is subject to strong lateral movement of suspended surface sediments. From left to right, they are selected NMR spectra of POC at 1,000 meter, 2,000 meter, and 3,000 meter at the same period of time. And as we can see, we do not see substantial change among the depths. Probably suggest non-selective preservation of POC during its sinking, as suggested by Hedge et al. Before. But we did observe a strong seasonal change for the shallow depths at 1,000 meter. The spectra for the March 2007 is dominated by LQ peak, which is probably driven by the increase in the lipid content. And this is only showed in the 1,000 meter depths. Okay, we also obtained all three depths POC samples at various times for East Japan Sea site. The East Japan Sea site, the water depth is much shallower with around 2,370 meters compared to more than 3,000 at station W. Again, we see the similar seasonal change at shallow depths at 300 meters. The March sample is dominated by the LQ peak and rich in lipid contents. And for the rest of the spectra, they all look similar as we observed for the station W. Okay, let's move to station M. Station M is one of the most important long-term abyssal time series sites in the Northeast Pacific. The water depth of station M is more than 4,000 meters, and it's known station M has very limited terrestrial input compared to the station W. We're only able to obtain the POC samples from just one depth at 3,450 meters. And the spectra at this depth are similar to the spectra I showed in the previous slides at shallow depths for station W and East Japan Sea. Probably again suggest non-selective preservation mechanisms for the POC. And there's not much seasonal change among the spectra. And we did not see the spring event we observed for the other two sites at shallow depths. Okay, we further did the molecular mixing modeling for the POC samples in the surface sediment trap for both station W and the East Japan Sea. where the spring event occurred. And the results showed that the lipid content of the March sample are doubled for both sites, as we can see, 34% compared to the around 17%. The content to March sample is almost double of the compared to the summer and the winter samples. And we speculate this event is caused by vertical migration of zooplankton, such as copepods. Copepods daily migrate between 200 meter and 1,000 meter, which is consistent with where we see the changes occur. In addition, copepods population is most abundant during the springtime, following the peak of the phytoplankton. The time is also consistent to when we observed these changes during the springtime. And this is the first direct observation of lipid pump contributes to the POC flux. I think we think it's pretty exciting. Okay, as I mentioned earlier, LQ-OLQ ratio is a well-established index for organic matter decomposition in terrestrial settings and has not been tested in marine environment. This figure shows the time series of LQ to OLQ ratios for station M at 3,400 meter and station W and East Japan Sea for all three depths. And we do not see any obvious trends among the depths, suggests that probably the LQ-OLQ ratio does not reflect the decomposition state in marine environments. And the large seasonal change we observed for the surface sediment samples for both station W and the East Japan Sea are ecologically driven by lipid pump, not a degradation. Okay, this figure shows the LQ-OLQ ratios of all the POC samples we received from all the sediment sites as a function of water depths. Obviously, we do not see the general trend of the marine POC does not follow the terrestrial setting with ratio increase with time. Instead, we see an opposite trend. It looks like we see higher and more valuable LQ-OLQ ratios at shallow depths and lower and more homogeneous LQ-OLQ values at depths. And the large values at the surface is, again, ecologically driven by lipid pump. Okay, the molecular mixing modeling results suggest a complex variation in POC chemical composition, possibly driven by multiple and different mechanisms. The x-axis shows the relative abundance of each biochemical class to the total carbon in POC. And the y-axis is the water depths. For the POC from all depths, it contains some degree of char and lignin, which indicates that all POC samples contain some terrestrial. I want to mention, both of the char and the lignin are terrestrial derived components, which means all the POC samples contain some terrestrial source, probably due to the adsorption of land-derived dissolved organic matter. And for station W, the green color, it seems the lignin content increases with depth. This is consistent to the strong lateral movement of resuspended surface sediments that are rich in terrestrial materials. And for the protein, it seems the protein content are much higher for the East Japan Sea samples. We think it's probably due to the unique geological locations, which has rich nutrients input. Okay, to conclude, no substantial changes were observed in chemical composition of POC with depths, implying non-selective preservation of organic POC during its transport from surface ocean to the depths. And we found the LQ-OLQ ratios do not solely reflect POC decomposition state in marine environments. And the global patterns of seasonal variation in surface POC organic geochemistry is likely ecological driven by lipid pond. And the increasing of lignin contents with depth at station W suggests lateral movement of resuspended surface sediments. I would like to thank Tim and Charlotte from ETH, Alan Jaffa from UC Irvine, and Mink Kim from Kyongbuk National University, South Korea, Liz and Hillary Klaus from University of Miami for sharing these valuable POC samples with us and for their insightful discussions. I also would like to everyone in Masayo's group at Rice University for their support. And with that, I thank you for your attention and I welcome questions. Thank you.
Do we have any questions online? So could I ask, what is the significance then that the POC doesn't degrade as it sinks? Is that what I'm hearing?
I think they are non-preferentially degraded, eaten equally by the microbes. The preliminary data suggest at least.